Thanks very much to the organizers for inviting me. So the first question you should be asking is, well, notice that I'm from a chemistry department, so what were the organizers thinking inviting someone from a chemistry department to the Cell Science Symposium? So my lab is really interested in understanding how cells process signals and how individual biochemical reactions are regulated to produce complex phenotypes that we see at the cellular level. So we're going to zoom way in and think about individual protein reactions, but all in the context of trying to understand how they function in the context of a cell. Before I get started, I want to acknowledge the two grad students, Maire and Erin, who did all the work. I don't do experiments anymore. I wish I did. Whenever I go into the lab, they laugh at me and take pictures, so I don't get to do experiments. Okay, so the big question that we're interested in at a lot of levels is how do cells process signals? And we know that cells can sense all sorts of different signals like growth factors, environmental stresses, hormones, and produce outputs like proliferation, stress responses, or differentiation. And cells use networks of proteins to respond to these signals. So an input signal will activate a cascade of signaling proteins that eventually leads to some output. Now, one of the big questions that we have about cell signaling is how do these cell signaling proteins find the right target in the cell? This is complicated on a lot of levels, but one of the real challenges has actually emerged thanks to the genomic and proteomic era. They've done a great job of identifying all these proteins and interactions that are important. And one of the fundamental things they've taught us is that a lot of these protein networks are highly interconnected. So there are signaling proteins that are shared between multiple different pathways. So our question is, how does this signaling protein know who it's supposed to be talking to downstream? How does it know who talked to it upstream? How does it know where the signal is supposed to go? How do cells repurpose these multifunctional components to perform unique and different functions? Now, another thing that you should be thinking, again, since we're at the Allen Institute, this representation of a cell does not meet the standards of the Allen Institute in any way. And that's actually important. We know that cells are not just bags of proteins. Cells have structure, cells have higher order organization. And so one way that cells can address this sort of problem is by using higher order organization to co-localize components. Use organelles to bring proteins together, co-localize things to different subcellular compartments. What I'm particularly interested in is another way to accomplish essentially the same task. So we also know that cells use these proteins that we've called scaffolds. These are proteins with multiple interaction domains that bind components of a single signaling pathway and bring them together. And so we link all the components of the signaling pathway together. And the idea here is that by bringing all these proteins to the same molecular platform, you're co-localizing them, you're making these reactions much faster than they otherwise would occur, and preferentially driving reactions down one particular pathway. So we can sort of think about this as a way to define a wiring diagram of the cell and specify input-output connections. So that's fine, but I'm talking to you kind of in cell biology language. As a chemist, what I want to know when I hear something like that, what does it actually mean to say that the scaffold is a wiring diagram of the cell? At the molecular level, what do we mean? How is it actually affecting reactions? And how can it define these connections in a way that prevents crosstalk between signaling pathways? So we'll come back to that promote question in a minute. I want to talk a little bit about the regulatory logic here. So how could we think about how a scaffold might help define specific connections in the cell? One way you might imagine that works, so say we have this shared kinase, and it can receive all sorts of different input signals and produce different outputs. If you imagine that the scaffold protein itself can undergo some sort of regulatory step. So the scaffold is normally inactive, so that kinase can receive its signals and do whatever it's doing to these proteins down here. But if you send a signal that specifically activates the scaffold protein, that scaffold protein can assemble this complex and direct the signal now to a new output. So this is a way you can imagine using regulation not just of the kinases, but of the other components of these macromolecular complexes to define signaling connections. And this is really, so two important things about this. One is that this picture of scaffold-mediated cell signaling comes from some biochemistry work that I and others did in yeast a couple years ago. We have a really good handle on how this works for some simple MAP kinase models in yeast. What we don't know is how this sort of mechanism applies to more complex signaling in human cells. So that's where we're going to go next. But why should you care? So we think about how are these scaffold proteins regulated. An implication of that is imagine that we're doing some sort of therapeutic intervention. Let's say this kinase has gone bad and started cancer, done something horrible, and we want to stop it. One challenge with all of our therapeutic interventions and kinase inhibitors is that these kinases are multifunctional proteins most of the time. Delivering inhibitors wreaks havoc all over the place in the cell. We would really like to have better ways to intervene with precision to inhibit just subsets of pathways, just the molecular functions that we want. So we need to understand, again, how are these kinases that are shared in multiple pathways repurposed? And if we can figure out these regulatory mechanisms at, say, the scaffold level, we could start thinking about how to intervene with precision. The other reason you should care is because we're starting to think now as synthetic biologists that we understand cell signaling well enough to go in, engineer, rewire signaling pathways, let's say take T cells and redirect them to new targets and do all immunotherapy and all sorts of tissue engineering, all sorts of cool stuff that really depends on us actually understanding what's going on at the level of these regulatory steps. And I think we're close, but we're still missing some fundamental things, as you'll see as we go through the talk. Okay, so I said this model of regulation comes from yeast. We want to think about whether this sort of thing happens in mammalian cells. And the model system that we chose to think about is Wnt signaling. We chose Wnt signaling for a couple reasons. One, it has multiple scaffold proteins, and it has a kinase that it plays a central role in multiple different pathways. So let me walk you through the basics of Wnt signaling, and then we'll talk about how it fits in the larger context. So the Wnt pathway you can think of, it's kind of a weird pathway in a lot of ways, not the least of which is that the Drosophila geneticist got to name everything. So the proteins have really funny names, like this protein disheveled because the fly was disheveled after they knocked it out. It's just weird. Anyway, some of these other proteins have Drosophila names that they have lost through history, thankfully. This is now a kinase. Sorry, I get sidetracked on this stuff. Anyway, okay, so what's happening here? So there's a kinase, glutathione synthase kinase, that phosphorylates a transcription factor, beta-catenin. In the off state of this pathway, this reaction is on. Beta-catenin gets phosphorylated. That phosphorylation event is a signal to degrade beta-catenin and keep it out of the nucleus and keep it from activating its downstream targets. When the pathway gets activated, through this receptor. So this Wnt ligand binds the receptor. It talks to this protein disheveled that made the funny looking flies. This protein talks to this protein that we think of as a scaffold, Axin, that is an assembly platform for GSK and beta-catenin. And somehow through the activation of this pathway, it turns this reaction off. Beta-catenin no longer gets phosphorylated. That means it accumulates, builds up, goes to the nucleus, and activates transcription. So, oh, sorry, went too fast. All right. So why do we care about this? What's going on with this scaffold? So the reason this is interesting in the context of the questions I raised before is because GSK is one of these multifunctional kinases. Its name, glutathione synthase kinase, comes from the fact that it was originally discovered as a regulator of sorry, did I just say glutathione? I should not have said that. Glycogen synthase kinase. Glycogen synthase kinase regulates glycogen synthase, which controls glycogen synthesis. So this is a kinase that plays a central role in insulin signaling. It also plays a central role in the Wnt pathway, which controls cell fate decisions, both through this sort of inhibitor of an inhibitor mechanism, where by turning off GSK, you activate this downstream function. All right, so how can we fit all this together now with our knowledge of, a little bit more knowledge of how the pathway works? So if what's happening in the Wnt pathway is that in the off state, this scaffold Axin is bringing GSK and beta-catenin together to promote the phosphorylation reaction. And then something happens when the pathway gets activated to block this scaffold, inhibit or inactivate that scaffold, and turn this reaction off. You can imagine a mechanism that will allow you to independently regulate Wnt signaling without necessarily perturbing whatever is going on here. And we could talk about this later, but you can imagine independent molecular mechanisms that would allow you to control activity through this pathway. Wnt itself would be specified by directly regulating the scaffold, not necessarily regulating the kinase. Okay. So now when we think about how this pathway could be regulated, we need to think first, okay, what does it mean to say a scaffold is bringing things together and making the reaction faster? So our simple model is that we're essentially making an intermolecular reaction intramolecular. So we're bringing things together and positioning them just right to reduce the entropic barrier for the reaction. And that's going to make this reaction fast. There are more complicated models. There is good evidence that some of these scaffolds actually allosterically interact with their targets to cause conformational changes that modulate kinase activity or substrate accessibility, and even more complicated things. But sort of the basic idea we need to think about for right now is, okay, so the scaffold is bringing these things together and somehow making them react faster. Then we can think about, if these models are right, how would you actually regulate the activity of the kinase? You would directly regulate Axin either through, say, binding partners that cause conformational changes or post-translational modifications. If Axin is a tether that's positioning things just right, then you can imagine perturbations to the structure of Axin to pull these proteins apart, and an inactive conformation could, again, tune the reactivity of this complex. You could also imagine that by tweaking Axin in some way that I didn't really draw properly, by conformational changes or post-translational modifications, you could disrupt the binding site and block the assembly of this complex. Again, regulate the activity of this kinase without necessarily doing anything to it. Okay, so how do we want to figure this out? So in my lab, we really want to understand what's happening at the level of these individual reaction steps. So how fast is this reaction? And how do we perturb it to make the reaction slower? So the approach that we take is to biochemically reconstitute the system, purify the proteins, assemble a minimal system, and see if we can understand what's happening. And then the key step that we're not actually going to get to today, because it turns out this was really complicated, is we're going to start perturbing Axin, do mutations, post-translational modifications, bring in extra binding partners, and ask what does it take to perturb Axin in a way that does something to block the reaction. So we want to biochemically reconstitute this and see if we can understand what makes it slow and what makes it fast. And it turns out other people have thought about this problem before. So these proteins were discovered 21 years ago. People have been thinking about this question for a long time. So one thing you actually should be thinking is, why are we still doing biochemistry in 2018 or whatever it is, almost 2019? It turns out these are hard problems. And we're going to end up, you'll see why in just a second. But there is a paper from back in 2003, so 15 years ago, that did essentially what we want to do. They purified these proteins, they measured reactions without Axin, and then they added purified Axin protein, and they said, we see the reaction get 24,000-fold faster. That's huge, that's amazing, and that's really interesting from a mechanistic perspective. We really want to understand what do you do to, or what is this protein doing to make that reaction so much faster? And so the first thing we want to do is ask, can we actually reproduce this number? Before we even started this experiment, and we'll again come back to this, it turns out that there are some weird red flags going on in this paper. The most important thing to recognize as we start going through this is they're never actually really clear about what reactions they were measuring. They never actually carefully defined a kinetic scheme and measured rate constants. Now, I know I'm talking to a cell biology audience, and you guys are saying, I don't care. It's going to be important, so let me explain why it's important. So this reaction can get really complicated really fast. So we're thinking about a three-component system. We have a scaffold protein, a kinase, and a substrate. The scaffold could bind either the kinase or the substrate, and then it needs to bind the other thing. And the model is that once it's in this ternary complex, it can react. The kinase can phosphorylate beta-catenin. Eventually, this complex falls apart. It can reassemble and keep on going through this catalytic cycle. So already this is complicated by the fact that there's multiple binding events. It's a little bit tricky to work out exactly what's happening. Things get a little worse in this scaffold field, and this sort of problem comes up in a lot of different multi-protein complexes. If you have way too much scaffold, you can imagine a scenario where in a huge excess of scaffold, you're going to have these segregated complexes where a scaffold will bind one of the partners, and a separate scaffold molecule will bind the other partner, and they never come together. So this is a problem. We need to be careful about the actual concentrations that we're using in these reactions. We have one thing going for us, and that is that people have measured these binding constants. We know that the binding of Axin to this kinase is actually quite tight, 50 nanomolar. The binding to beta-catenin is quite a bit weaker. And that means we can make a simplifying assumption. In our limiting case, we can imagine conditions where we can work at concentrations where Axin is binding the kinase. And we'll start out with beta-catenin not bound to the scaffold. By varying the concentration of beta-catenin, you'll see in a second, we can measure this bimolecular rate constant kcat over km, and we can measure kcat, the unimolecular rate constant, from this assembled complex. All right, so now we got to figure out how we're going to do this experimentally. Oh, I'm sorry. And then, so this is the scaffolded reaction we're going to measure. We're going to compare it to the corresponding rate constants for the unscaffolded reaction. So the comparison states we want are the bimolecular reaction for just kinase to beta-catenin, and then again, the unimolecular rate constant for this assembled complex. This bimolecular rate constant is going to tell us things about whether binding and assembling this thing in a ternary complex in the transition state is actually, if Axin is actually helping to assemble that ternary complex. The unimolecular rate constant might tell us something about whether assembly of this complex perturbs the structure in a way that makes the reaction faster. Okay, so how are we actually going to do this? So again, this is the system that we're trying to model. This is what, or the minimal picture of what we think is going on in the cell. In my lab, we're trying to make it even simpler. We want to purify these proteins. We're going to express them in E. coli from recombinant sources, so we hopefully know exactly what we're getting. The Axin glutathione, I just said glutathione again, that's not good. Glycogen synthase kinase, and beta-catenin. And then we're going to do some quantitative kinetics. The way we do our kinetic assays, so we add ATP, we do in vitro phosphorylation, and we're going to measure phosphorylated beta-catenin using a quantitative Western blot. So we take time points, each one of these time points, measure how much beta-catenin is formed. I'm going to give you a quick and dirty kinetics reminder because I know a lot of you haven't thought about this for a while. So we're going to measure phosphorylation of beta-catenin as a function of time, and the slope of this reaction is the initial rate. Now we're going to take each one of these, measure these initial rates under multiple different conditions. So we're going to vary the concentration of beta-catenin, sort of this first order expectation, more substrate reactions should get faster. Now here's what happens when we actually do the experiment. So we vary the concentration of beta-catenin. Each one of these points is an initial rate for a separate reaction. And we see what looks like standard Michaelis-Menten kinetics. Now the error bars are quite large. These reactions are really slow. That's okay. We expected the reaction to be slow. The data's a little noisy. We can fit it to a Michaelis-Menten model. And this part of the curve tells us about the bimolecular rate constant. This is the unimolecular rate constant. We don't care what those numbers are. What we want to know is, do these numbers change when we add Axin? Okay, it's a lot of build up to this point. We're going to add Axin. And what happens? Nothing. This is really troubling because that paper told us that it should have gotten 24,000-fold faster. Did we screw things up or did they screw things up? We'll find out. Okay, so maybe we're not working at the right concentrations. You know, we did pick our concentrations carefully. Let's vary the concentration of Axin, bump it up a bit, and see if there's any hint of an increase in reaction rate. There's no hint of anything happening here. So we add a lot of Axin. We're still working under conditions. So this is the binding constant for Axin binding to GSK. In this whole region of the concentration range, we should be forming this complex. We have separate evidence that we're forming this complex. We're not quite all the way to saturating this binding constant, but it doesn't matter. If there was a 24,000-fold rate effect, we should have seen something out here. There's not a 24,000-fold rate effect. Okay, but the last thing we did to try to figure out what is going on here, we need to figure out what's happening. And we did one last important control that every enzymologist needs to do, which is vary the concentration of the other component in the system, vary the enzyme. Expectation is that as you increase the enzyme concentration, the reaction should get faster. It should scale linearly. This should freak you out right now. You guys should all be terrified. This is not good. This is what was supposed to happen. As you add more enzyme, it gets faster. Instead, what we're seeing, and this is the reaction without Axin, for some reason, it's leveling off in a really, really weird way. This is deeply troubling. We stared at this for a long time. We cried a bit. We did the experiment with Axin and saw something strange, which is that in the presence of Axin, it actually behaves linearly. So we're actually using a big excess of Axin here. And we vary the concentration of the enzyme, and it scales linearly. So at these high concentrations of the enzyme, it does look like empirically there is an effect of the scaffold. You add the scaffold, the reaction gets faster. All the data that I was showing you before was done at these low concentrations, actually in the linear range of enzyme concentration. So we're not seeing an effect down here, but something's happening at high scaffold concentration. And our simplest model is that actually what's happening is that this kinase is coming together in some sort of higher order structure that's inactive. It needs to be in this monomer state to react. And so what we're actually seeing is this inactive state. When you add Axin, it's driving it to this monomeric state that can then react. We're not actually having any effect on these rate constants as they're defined in this scheme. What's happening is when we do the reaction without Axin, we're just getting stuck in this inactive state. So you're not seeing apparent reaction because the kinase isn't behaving. It does actually increase empirically the observed rate, so that's interesting. Maybe it's stabilizing GSK. But the take-home message is, okay, so Axin does accelerate the GSK reaction, and we can see this in vitro. It's not clear at all to us yet if this model of GSK stability matters in vivo. Well, let's not worry about that and just take a step back and think about what's going on here. Where did this model come from? Why has the field for 20 years thought Axin is a scaffold that brings proteins together, and that's how this pathway is regulated? So there's some really important and valuable cell biology that was done. So the first couple experiments were, okay, let's delete it and see what happens. And if you delete it, what happens is you break this phosphorylation reaction. It acts as if the pathway is on because this reaction doesn't happen anymore. That tells you the scaffold is necessary, but it doesn't actually tell you what it's doing at the molecular level. Then we have this paper here that sort of drove us off a cliff. that told us that this reaction should be 24,000-fold faster. And if you go back and look at that carefully, what you find is that what they actually did was they observed through a single time point comparison of two different reactions, a 40-fold effect, never did any of the sort of kinetic analysis we're talking about. So rate constants matter, and I think maybe this is one of the take-home messages. Looking at the difference in intensity between two bands on a gel is not enough to tell you something about how reaction rates change. You actually need to think about doing these experiments in a biochemically rigorous way. And I'm going to wrap it up real fast. Okay, so the take-home message is not as exciting as I led you to believe, because I led you to believe we were going to learn something cool about how this protein was regulated to control kinase activity. Instead, what we've learned is that we're missing something really fundamental about this pathway, but that's important. We've learned that our minimal model, this Axin by itself, is going to bring these kinases, the kinase and substrate together, is not sufficient to activate the pathway. There's a lot of other interesting stuff that could be happening here. There's other kinases involved. Those of you who are familiar with pathway know there's a priming kinase called casein kinase. There's this protein in gray that I haven't even talked about called APC that is known to be important in the pathway. Mutations cause cancer. It's bad. It also appears to interact with axin and beta-catenin. So our favorite working model right now is that the scaffold actually is this complex of two proteins, axin and APC. We have a hint of preliminary data telling us that might be what's happening. And we're going back to the biochemistry drawing board. Can we purify the proteins? Can we build a minimal system with purified components and find out what it takes to accelerate this reaction and regulate this reaction? Okay, yeah, so we already covered that. So thanks to Erin and Maire who did all the work, and these institutions for funding, and thanks for your attention.
We have time for a question or two.
I'm curious to see how you determine axin folding, because you're doing, bacterial expression.
Yeah, so we don't have any, so the question is, how do we determine axin folding? We don't actually have any rigorous way to do that, and that's a general problem with these biochemical reconstitution experiments. So we can do things like measure binding constants and try to reproduce known binding constants that people have previously measured. They're faced with the same problem. Is their axin fully folded? So we don't actually know for sure. We know, we can build a biochemical complex that is capable of binding both of these targets. And so we know that much at least. So in our in vitro conditions, it can bind both of them. That's good enough. I mean, what the absolute numbers are, we don't necessarily care about. I think way back there.
Hi, really neat talk, thank you. I was wondering, about what you think about the general question of how general do you think it is that these scaffold proteins isolate, individual pathways from crosstalk, or versus, how deep does the rabbit hole maybe go where there's further regulation on top of that, and we find out there are more interactions, more crosstalk that's scaffold mediated? Yeah,
so that's a great question. So are scaffolds really insulating pathways? I think, so the answer is yes to all of the above, and that's kind of a cop-out answer, but that's the real answer. I think there are definitely examples where the crosstalk actually matters, where you want to activate a shared kinase and you want it to branch out and hit multiple targets. There are other pathways, the one we worked at in yeast, where it was very clearly important that the pathways needed to be insulated from each other because they essentially drove two different cell fate-like decisions in yeast. And in that system, it turns out that, yes, that molecule we called a scaffold was really important for insulating the pathway, but the mechanism by which it worked had nothing to do with co-localizing proteins or tethering them together. It turned out to be really complicated allosteric conformational change that had membrane recruitment in the mix. So I think the take-home message is these things we call scaffolds, these accessory proteins, they're important. There's no doubt about that. You knock them out and bad things happen. But our understanding of what that actually means in molecular terms is still pretty weak.
Thank you.
Great. Thanks so much to the speakers from this section. So we'll take about a 15-minute break and resume back here at 2:35 for our last groups of talks for the day.